Thank you, colleagues, teachers, young students, all of you who are here. And I'm really very delighted. This is the SEP program is one of the flagship program of Indian Academy of Sciences, and including the teachers training program. And I was very fortunate to host so many of them in the lab. A couple of you are here in the audience. It was always a very vibrant feeling when the young students come to the lab and spend a few months in the lab doing some excellent work, and some of them did so well that they got the second term of the SCP. So the teachers and with them also we had a very different kind of a dialogue. So what I'm very happy about is this audience is just not a biology audience. We have our teacher colleagues who are from physics, chemistry, and mathematics, and I always currently I am also the UG Associate Dean, and I always tell that if somebody wants to do a good biology, one must have a very good basic foundation in physics, chemistry, and mathematics. And that is where the crux lies of asking a question. So why I'm very happy, because the audience here, the teachers are from different backgrounds, and some of you, my young students who are sitting in the crowd, they are also from a different background but having a common theme of focus in understanding the life form, that is a biology. My lab focuses on infectious disease, so I am a medical microbiology by training. Throughout my tenure in the career, I worked with infectious disease, trying to understand that how such a tiny bacteria or a virus can actually make us ill. With few thousands of gene to 30,000 gene in the body, what that we do not have, that we are still not able to answer a simple question of how to take care of infectious disease. The entire world is crying with antimicrobial resistance. You open a page of anything, be it CDC, WHO, newspapers, everywhere the cry of antimicrobial resistance, death is increasing, so etc. etc. So today I'll take you through an interesting story of a bacteria, and that is how the title is Life in a Vacuole. So the pathogen of the choice is Salmonella. Just for the crowd who is not much aware of the bacteria, this particular pathogen, Salmonella, is a pathogen which affects the body through orophycal route of infection. That means if you take contaminated food and water, and if the bacteria is present there, it will get into the gut. It will, in fact, be able to take care of the acidic pH of the stomach and then land up in the small intestine. Now, pH of the stomach is roughly around 2 to 2.5. Anything that is there should be actually chewed up. But these bugs are so smart that they have their signaling mechanism to bypass that acidic pH and then finally end up in something called as the small intestine. So in the small intestinal lining, if you still remember your 9th class biology, the small intestinal lining is not naked. It is lined by very thick mucus, which is around 150 micron thick mucus. And on top of the mucus lies your gut flora. What we call it, as a second brain, a very big group of research on the so-called gut microbiota or the microbiome. I'm sure that you all must have heard of it. So this microbiome protects us. But if that is the case, how these Salmonella can enter? That was a question that was a mind-boggling. You have a thick mucus. On top of that, you have a microflora, 230 million different species. It's like a mat. So how can some 100 bacteria just come and enter our system? System is so well guarded, but it happens. We suffer from systemic typhoid fever. We suffer from gastroenteritis and diarrhea. So what they do is, the nature is sometimes very interesting. Though you have a very thick the thick mucus lining, some of these cells, like dendritic cells, like a M cell, they throw their arm in such a way that they rise above the mucus and above the flora. And these bacteria, they beautifully come and slip inside. It is like just sliding and slipping inside the hole. And that is how their systemic intervention happens. So they get inside the system, and they can be transcytosed from the apical side to the basal side, and from there they disseminate into various different body organs. That is how this bacteria causes the disease. Now, why Salmonella of all? According to the WHO, this had been put as a high priority pathogen. till a couple of years back, this was in bioterror category number 2. But due to the devastating effect on the morbidity and mortality, this has been put in bioterror category number 1. So this is a pathogen of concern. Anybody who is not very careful with the food and water, please try to become very careful, because you do not want this bug to get inside your system. It is a debilitating disease. It's just not some kind of a fever and you get cured. There are long-term implications of someone who actually gets this bacteria inside the body. Now, what happens when it gets inside and it gets disseminated? It resides in this cell type. Now, this cell is known as a macrophage. Macrophage is a cell in a body which should clear anything that gets inside. It should take up and it should get destroyed. But what they do, this bacteria, they will not allow them. But on top of that, the bacteria gets inside and they rearrange their entire structure of the macrophage and act them as a vehicle to go from one site to the other. And all this is done, we will discuss about it, is done by a certain gene in the bacteria, which we call it as a pathogenicity island. These are the big chunks of DNA which is acquired by horizontal gene transfer into the bacteria, which gives them a fitness advantage. And I call them as an amazing secretion system injection supramolecular needles. They use this needle structure to deliver the effector protein into the cell to take it up and have their life. There is another pathogenicity island, and this was my own work when I was a postdoc, that how this particular pathogenicity island will allow them to replicate. Now, why do I call it as a life inside the vacuole? I'll come to that. Now, Salmonella is an intracellular bacteria. What is meant by intracellular bacteria? Suppose a bacteria gets inside the body, but it does not want to enter any cells. You call these bacteria as extracellular bacteria. They will be floating around in the body. They may get cleared by the blood cells, or they may actually just float and go elsewhere. And eventually that can cause sepsis, or the patient will get rid of that. But intracellular bacteria are like Trojan horses. If you have read the story of Trojan horses, that how very sneakingly the bacteria gets inside the cell. Not only that they get inside the cell, suppose now this is an example, this is a beautiful experiment done by my colleague Sandeep Ishwarappa. In fact, he is the first PhD colleague with whom I started my lab when I joined IISc in 2004. Sandeep was a medical student, and he was my first PhD student also. And he very patiently used to infect the cells. Currently, of course, he is an associate professor in the department where Professor Ganesh is, that is in biochemistry. And he was someone who had such a patience to look under the microscope to figure out that where this bacteria actually resides. You see here, this is a cell, macrophages, which is labeled with the antibody to RAP7. Now, RAP7 is one of the protein which can mark the Salmonella containing vacuole. And why do we call it as a vacuole? Because each Salmonella, if you see here, the green is a GFP expressing Salmonella. Each one of them, they stay in a single compartment. Not only that they stay in that single compartment, which is bound with a boundary, they never leave that compartment. Now, can you imagine very stringent life of the Salmonella? It is in a very, tightly bound compartment here. This green bacteria is staying in a tightly bound compartment. This is just one of the marker for the Salmonella containing compartment. There are 40 proteins from the host and some from the bacteria which makes this house. And they don't leave the house. Then how do they survive? It's not that it is very spacious compartment and then they get enough food, etc. On top of that, the pH of this compartment is acidic. It is not even a neutral pH that they can survive very well. So stringent pH, low nutrition, absolutely no way that it is very luxurious. Then how do they survive and how do they become so successful? So what was figured out, so you look at this EM structure, what it shows is this is indeed in a vacuole. This is a Salmonella. Just look at this. This is a rod-shaped bacteria, by the way. Salmonella is a rod-shaped gram-negative bacteria. And it has a peritrichous flagella. That means it is a flagella all over so that it can move. And of course, it's a gram-negative. It does not form spores.
So when you see there, see this is a Salmonella. Of course, it depends. It's appearing to you as a round because the electron microscopic section was cut in that particular plane. So it is bound in a vacuole, and what is very striking about it is the bacteria, once it enters, will it only stay there? No, right? There's a right to live is the right to reproduce. So it will reproduce. Now you know about the bacterial division. So it's a binary fission. One bacteria into two, two into four, it's a logarithmic growth. So in this case, if this bacteria has to divide, what is the status of the vacuole? Will it come out of the vacuole, divide and then go again? So that was not the case. In fact, the Salmonella divides along with its own vacuole. So the bacteria divides and the Salmonella containing vacuole also divide, which make them to come in a single Salmonella in a vacuole. And this was something very interesting. So how bacteria divide along with the vacuole was a question that is still puzzling us. It's 2 decades in the lab. Some wonderful work has been done, and not only that, this particular bacteria is an example of the first example of something with such a tight-fitting vacuole. You do have like Legionella. They live in a big spacious vacuole, big home. This is the one which is a very stringent vacuole. And if it comes into the cytosol, the life will be much simpler. But no, they don't want that. They stay within the vacuole. The question is, how do they divide along with the vacuole? That is the question. So the first thing is that how many bacteria does the Salmonella containing vacuole harbor? That was a main interest. If it is a big vacuole containing multiple Salmonella, then what happens is the bacterial number goes down and the host wins. That means if it's a big vacuole with many bacteria inside, the Salmonella dies and the host wins. But if it is a single bacterium per vacuole, then the host dies and the bacteria survive. That means that the single bacterium per vacuole or the vacuolar lifestyle is a strategy for the bacteria to establish successful pathogenesis. Now, bacterial fission is okay. So there's a lot of beautiful mathematical dynamics that plays a role here. I mean, we have done a beautiful modeling to understand that what is the capacity of a cell to harbor how many number of SCVs. First question, what is the threshold that the bacteria can undergo how many generations to have the cell? Is it something that when it is filled up, will the cell burst and can it then reinfect the population? And if it does it come along with the vacuole? So the secondary infection will be the bacteria along with that compartment coming out and infecting. Then what will be the dynamics? The dynamics may be something much more interesting than a naked bacteria which is coming from environment. So all of them extremely interesting questions that kept us really busy. But we scratched our head, and how do you take this very surprising fact that Salmonella containing vacuole is dividing? Because vacuole is made up of host protein, all the host protein. What initiates the cues for the division? So we took some very good, a cue from mitochondria. As you know that mitochondria is a good example of an endosymbiont, and it is none other than alpha proteobacteria, which once upon a time had harbored the cell and it became one of the important organelle, and mitochondria also divide. So DRP1 apparatus, this is one protein which is important in making a site of mitochondrial division site to give rise to 2 mitochondria. So we asked a question, does the Salmonella containing vacuole also uses the same BRP1 machinery as it uses in a mitochondria to make this bacteria to get into 2 vacuole? And indeed yes, partly yes, because when you treat the cells with the SOV, sodium orthovanadate at a particular concentration is going to inhibit the DRP1. So if you do that, what we see is the multiple bacteria per vacuole increases, and with the multiple bacteria per vacuole increasing, the intracellular bacteria or the life of the bacteria within the cell goes down. That means the Salmonella containing vacuole must be a single bacterium per vacuole, and if that is perturbed, then the bacteria will meet with a dead end. Now, my chemist friends here, you will be the best one to actually, ask a very important question. It's a multi-billion-dollar question. that can we find a small molecule inhibitor which is going to inhibit the vacuolar division. You don't worry about the bacterial division. If the vacuolar division is perturbed, the Salmonella cannot survive. How much ever number big bacteria, vacuolar bacterial growth you have, the bacteria will not survive if they come in a multiple bacteria in a vacuole. And indeed, it is true if you also prove it in another way. Say, for example, if you use P50 dynamethene, which kind of quenches the DRP1, you see that all the bacteria, they come in a one vacuole. So you have again a multiple bacterium per vacuole because your DRP1 action is somehow inhibited. This is a control where you see that, yes, there is a single bacterium per vacuole. When you have this multiple bacterium per vacuole in the DRP1 condition, and even in the INT4 cells, which are the epithelial cells, you see this. See this particular part? Multiple bacteria in a vacuole. And all of these bacteria, in fact, when you do their live and dead assay, they die. So they are not able to establish the infection. So we have a really amazing tool now to understand that don't tackle the bacteria per se, because it's a right to live. If we have a right to live, so are they. And if you perturb their life, that is what we are facing, the antibiotic resistance. So don't perturb that, but you think intelligently about how do you perturb something else so that you can at least manage the infection status. And we again figured out that this Salmonella is very unique in, perturbing this cell functionality. Wild type means it is a bacteria which is infecting the cell, and the moment the bacteria gets inside the cell, all the lysosomes, which are supposed to be the end fate organelles to take care of your exogenous life forms, to clear them off, all these so-called volume of acidic compartment which you see in the uninfected cells are completely gone in the infected cells. It can mean two things. First, the bacteria may secrete a factor which may neutralize the pH of the lysosomes, or it may somehow modify the expression pattern of the lysosome forming gene, that no lysosomes are being formed. So what is it? Of course, by again very tedious experiments we figured out Salmonella does not neutralize the pH, but it sequesters all the cargo which is going to make the lysosome toward making its own Salmonella containing vacuole. So they make lot of SCVs and the lysosome numbers suffer. So there are not enough lysosomes to actually take care. This is a controlled concanomycin. Concanomycin is a VATPase inhibitor, and it is going to actually neutralize the volume of acidic compartment. The bacteria acts actually exactly like that of the VATPase. So to that extent, it neutralizes the volume of acidic compartment. And indeed, that is not only in the cell line. If you take a mouse which is infected with if you take a mouse which is infected with Salmonella and you harvest the spleen is an organ where bacteria goes. Yeah, there is something called as splenomegaly. The spleen becomes enlarged. You can take the spleen and you can just it's a soft tissue, so you can just tease it up with two glass slides, and then you sort out the cell and you see that wherever there is enough bacteria, the pH of that particular compartment drops. So it is indeed true that the volume of acidic compartment is significantly dropped by this infection, and this may be one of the very intelligent strategy by the Salmonella to establish its growth within an organ or within a tissue. So one more intervention strategy can be thought about. How do you not allow the lysosomal cargo machinery to fall? So you have to constantly upregulate the machinery so that the lysosomes can be formed to take care of the infection. And this would be a good intervention strategy. Now, to prove further that you need to really do, we came across something which is interesting. It's just not the mitochondria. Nowadays, as a cell biologist or as a chemist or as a physicist or mathematician, we are always constantly challenged by one factor, that how do you really prove that that is the case? It's so much in biology. It's just not a one-way contact. Now, if it's just not a mitochondria, because mitochondria makes a contact with the endoplasmic reticulum, and then it has some other contact with the lysosome. So now we are talking about a three-way contact. Here also, so it's just not the mitochondria we saw, but when you have the when you have the KDL sequence, which can mark the ER also marks the site of vacuolar division. So mitochondria and ER both are playing a role in marking the site of division. Now what puzzled us for a very long time is, we can understand that a bacteria divides, it's a binary fission. A 2 micron bacteria undergoes to give two daughter cells of 1 micron each, fine. But then what orchestrates the division of the vacuole? Because vacuole and bacterial division should orchestrate with each other. And they should really coincide to give rise to two bacteria in their individual vacuole. How does that happen? That orchestration is important. And after very tedious experiments, we figure out that even the ER comes and grabs around this Salmonella containing vacuole to give it a site to give it to two bacteria per vacuole. So yeah, there are a lot of these beautiful snapshots of the construct where you can see that, if you can see here, that this is a dividing bacteria and you see a constriction here, and this particular constriction is the one where this actually the ER is getting wrapped. We have some very amazing live imaging microscopy also to see the same. I'll just skip this for a moment, and these are all the additional experiments to prove that you have to do some dominant negative DRP1 and ask the question that what is important for the growth of the bacteria and for the division of the vacuole. And every single experiment proved that single bacterial per vacuole is required. It is a key for the growth. Now, as I mentioned to you that the intracellular lifestyle is very important, and you see here very clearly that this is a growing bacteria, this is the vacuole, and when the bacterial constriction happens, the vacuolar constriction also happens, which leads to two bacteria in a vacuole. Okay? Now this is quite clear. And we know that, and otherwise you see them all in the single bacterium per vacuole. You will never see in a cell multiple bacterium per vacuole until and unless you use some intervention strategy. So this is very clear. Of course, for mitochondria, it is an amazing endosymbiont, and we know that even if you talk about a lot of these cells as a organelle formation, then you work into the dynamics and you see that when the mitochondrial divide, how the division happens within the mitochondria. Now, coming to the ER, as I mentioned, that ER forms a very important point of contact. The ER stress is important to make the ER to change their status from a sheet to tubule. So ER actually also exists in many other forms, like it can be as a sheet or it can be as a tubule. So the Salmonella infection indeed lead to the increase in the ER stress. I'll show you how do we see, and why did we focus on ER much more? Because we realized that the moment you have a growing Salmonella, you need more of the membranes. Otherwise, from where are you going to draw the membranes from? And why the ER should be giving up the membranes? You know, so the ER should have a long tubule with which it should wrap around, like a rope, which is wrapping around any object. And it is indeed true that couple of the ER stress factors like ATF4, they are all increased upon Salmonella infection. Don't worry about this particular mutant. So in our lab, because we have a battery of mutant with all the pathogenicity factors, we generate the mutant in the lab. Now this particular mutant, SSAB, cannot secrete any of the spike effector proteins. So they are kind of attenuated strain. So whatever the wild type is doing, this particular mutant is not doing that, because they get killed, they get cleared by the cells. They are a very good control for us. And yes, it is indeed true that there is a stress involved. Then what you do is you go on counting the tedious experiment again, the number of cells, and you figure out that all these proteins, be it ATF4 or ATF6, upon infection over different period of time, the stress response is high. That is for sure that the stress response is high. Sure, with another protein that is the ATF6, again the number of counting shows that the stress response is very high. But then how does it implicate? What is the implication of this ER stress with what I was talking about is the division of the vacuole. what implication it has or what connection it has. Now, Salmonella infection, as I mentioned, it leads to the structural changes in the ER. And what are these structural changes? Now, this is a very beautiful schematic which shows that how there is a connection between the nucleus and the ER. These are all the proteins which makes these connections and the channels, and we concentrated. So this part was carried out by my, my very recent young colleague who graduated from the lab, Umesh. Reticulin 4A. That is one of the very important connection that is there to make these point of contacts. And it is indeed true that all these transcript level of be it reticulin 4A or CLIP63, specifically reticulin 4A and CLIP63, these protein increases in the wild type, not in the mutant, not in the only uninfected cells, but in the wild type. That has a significance. Why should it increase? I mean, why should reticulin 4A increase when the cells are getting infected? And we figured out that yes, it is indeed increased. So again, you have this KDL sequence, and you see that the KDL sequence is exactly marking the site of the dividing bacteria. So the percentage of the tubular ER increases dramatically. Sheet is of no use. So the sheet conversion to the tubules. Now we are trying to understand that actually how many tubules, what are the dimensions of the tubules and how many turns will it happen. So we have a very good collaboration with our physicists and the mathematician across. And in fact, they only gave a hint that don't think that because the bacteria is dividing, there is a suction force which will allow the vacuole to suck in. That is not the case. There is something which is happening due to the live bacterial processes. So you must define what are those live processes. It is not as mundane as a suction force which is giving you per bacteria as a vacuole. And that is how in fact we could do so many beautiful experiments. So yes, it's a tubular ER, and the tubular ER always increases in only infected cells, not in the bystander cells. The bystander cells means in a given plate you will have some number of bacterial which gets inside, but the other cells will be uninfected. Those are called as the bystander cells. Why do we have to do experiment with the bystander cells? Because there can be something like a signaling which is produced by the infected cell which can affect your bystander cells. And one has to always, if you are working with two life forms, a cell, a bacteria, virus, parasite, always make sure that your system will not have 100% infection. So you should look very carefully into the pockets where in a plate you have an infected cell surrounded by non-infected cells. So what happens in those non-infected cells? And yes, the bystander cells in fact have no effect. It is just like the UI. So whatever the sheet to tube conversion, tubular conversion is only there in the infected cells. So saying that it is definitely required for the expansion or for the wrapping around. And how does it play a role in the SCB division? That's the question. Yes, then you do experiments with all these, constructs which is going to be inside the cell, and then you look into the fold proliferation of the bacteria, which is a hallmark of a life. Indeed, when you transfect your cells with the reticulin 4A construct, look at the fold replication. They go very high. This is an empty vector, the normal fold of replication. Moment you have a tubular reticulin, the fold proliferation goes high, showing that the marking is extremely important. And when you knock it down, how important is reticulin? So you knock it down by using the shRNA, and you see that really the fold replication drastically decreases, showing that yes, it is very true that it helps in the SCB proliferation. Again, huge number of very tedious time-lapse imaging to show that the reticulin 4A is very important, or 661 is very important for the vacuolar division. So I will just see whether I can play this video. If not, this is one beautiful video which shows. Okay, so this video shows, if you really focus on this particular, this was this one bacteria, and you see here that how now this is getting wrapped, and then you have the two bacteria. So this live imaging, in fact, is clearly shows that endoplasmic reticulum forms a point of contact and it wraps around, which leads to two bacteria in a given vacuole. So the bacteria divides along with its vacuole, and this is what is an amazing finding that we got. And finally, again, did lot of other time-lapse imaging to show the same, that at what time point, look at, these are all the lapses, yeah, time lapses, and a different frame shot to show that indeed this bacterium gets marked by the endoplasmic reticulum tubules, and then they get into two bacteria. This all convincingly proves the same. Again, a huge number of experiments, other kind of softwares to show that how is a groove, how the wrapping is done, etc., etc. And we were not very successful in doing the film SEM because the instrument was not there in India. And we are trying to now, collaborate with Australian group and indeed do this film SEM experiment to prove. But other than that, you see a very clean marking or the imagination and a very clean division along the axis of the bacterial division and the vacuolar division. So yeah, so far so good. ERT views facilitate the SCB division, and if you compromise that, the bacteria comes in one vacuole and the bacteria dies, and there is no doubt in that. But then the question comes, what makes the ER comes into the site of the division? So till so far we know that the ER tubules marks the site, and if you have the less in the ER sheet, there is a less SCB inside the cell. So the tubules make the SCR sheet grow like this. It's beautifully marking the site of the bacterial division. But what marks the site? This was $1 million question which was bothering us, because it cannot be only from the host site. There has to be something from the bacterial effector protein which is attracting or recruiting the reticulin forae onto the site of the division. And we were lucky, in fact, to figure or stumble upon an effector protein which is called STEA. STEA is an effector protein or a virulence protein of Salmonella, which in fact is essential for the SCB fission. If you do not have STEA, so when we deleted the STEA gene, you see the number of bacteria per vacuole increases. So multiple bacteria in a given vacuole. Not only that, when you have a multiple bacteria, look at the STEA mutant, all in a big blob. And this blob could not survive because they get attacked by lysosomes very easily. It's a bulky vacuole, no? So they get targeted to the lysosome easily and they get completely cleared off. So we realize that STEA is the one which marks the site and which is important for the division. And so now we have an amazing model showing that it is the STEA, but the question still remains how the STEA brings the endoplasmic reticulum. That is still remaining. We could not figure out that what is the dynamics. STEA comes, it marks, and then the reticulin is wrapped around like a zipper model. Or it is the some other protein which is tethering the STEA with the endoplasmic reticulum. So there are a couple of more experiments which are required to be done. But without any doubt, if you have a mutant of STEA, they are not able to proliferate. And as you see here, the single bacterium per vacuole in the wild type where everything is intact and where the STEA is knocked out, it's a multiple bacteria that comes within the vacuole. So, and the SCB diameter also increases. We are now trying to understand the purifying these SCBs, which is again not a very easy task, to purify these bulky SCBs, and then you take this, analyze these individual SCBs in terms of its dimension and the content and the life inside. So let us see how much we will be able to do. I am going to just tell you that because they are now in a single big vacuole, bulky vacuole, they are very easily targeted by the autophagy machinery, and they are somehow getting cleared off by the lysosome, by the other autophagy machinery, by the very sheer of it's able to not decrease the lysosomal number. So the wild type, it is able to actually decrease the lysosomal number, but when you have this STEA, the lysosomal numbers are high and they target the large vacuoles. So it is indeed true that STM wild type can decrease the lysosomal number, but the STEA cannot. In a nutshell, if I really mark the summary here, It is very important for this particular bacteria, Salmonella, and I have no other examples of this bacteria. This is a very unique one. To live in a single bacterium per vacuole and never leaving its vacuole. They are quite happy, so the life in the vacuole is the key for their successful pathogenesis. How do they do that? They not only get inside the vacuole, but they have a very successful lifestyle by dividing its vacuole. So they have a very successful lifestyle by dividing its vacuole, and if they are perturbed in their division by either not having a STEA or not having the reticuli tubules from the host, they end up in this bulky vacuole, and these bulky vacuoles are not able to divide and grow, and that brings down the pathogenicity of a bacteria. So if we have to really think of a very smart intervention strategy to counter this disease of typhoid and gastroenteritis, we cannot discover more antibiotic. It's of no use because it will make resistant. What we need to target is how do you inhibit the vacuolar division, that is one, or how do you somehow inhibit the wrapping of the reticuli around the Salmonella containing vacuole. So these two can be an amazing strategies to counter this infection. I'm very blessed always with my group of colleagues, very vibrant colleagues, all in red in this column. They have all graduated and they are faculties or postdocs. These are the ones who are currently in the lab doing their PhD. So that is how I'm here. They are extremely vibrant. And because I am a microbiologist by training and I totally believe on the right to live, it's not that my somehow my passion and love for microbes. So this is what I wrote, a very small piece. This is a piece which I wrote for tale of antibiotics. This is one tale. This is a very small piece that I wrote for all of you. We are tiny. We are small. We need microscope to see us all. You need microscope to see us all. Still, we are mighty and will kill you all. Give us stress, give us pain. Still, your efforts will go in vain. We are community, strong and high. Treat us with respect and we treat you right. That's what is happening because of the antibiotic stress. We are not bad, but you made us so. We will become worse if you continue to do so. Hold back and keep quiet, and we will be harmony throughout our life. This is actually the harmonious conditions that we are all dreaming for, and hopefully we will do that in your future. Thank you very much, and open to questions.
Thank you, Professor Dipshikha, for an excellent talk, and then open for some couple of questions.
So, good afternoon, ma'am. I'm Srijit, and I do have 4 questions.
Yes, sure.
My first question is, apart from the STEA, are there any other possible factors that affects the that helps in the proliferation process?
In the proliferation of bacteria inside? So, good question. From our pull down, we could not actually find anything other than the STEA, which can make all the bacteria come within the vacuole, in a bulky vacuole. So, there may be some other factors which can aid in the vacuolar division. Proliferation, it will have, see, STEA mutant, they don't have any role in the in vitro or in the environment, they grow. But the moment they are within the cell, these bacteria are attenuated because they cannot divide their vacuole.
My second question is, how does STEA help in lysosomal trafficking?
Okay, STEA in general has nothing to do with the lysosomal trafficking. Only what it is doing is it is allowing the bacteria to somehow recruit the endoplasmic reticulum tubules to give 2 bacteria into vacuole. Now, the vacuolar structure or the cargo is made from the similar protein by which the lysosomes are made. LAMP1, LAMP2, others. So, all the cargo is getting sequestered toward the growing number of SCB. That is how you are getting a low number of the lysosomes. That is how indirectly STEA is playing a role because it is giving more single bacterium per vacuole.
So, there is no movement of lysosome towards the periphery region during infection?
No, the number of acidic volume or the number of lysosome itself is completely down. So there are no lysosomes. It looks as if you have treated the cell with a VATPase inhibitor, concanomycin.
My third question is, how does ER know where to wrap around the construction?
Very good question. That is what is because the entire. Now, how does ER know where to wrap around? When we looked very carefully, we realized that it is the ER tubules which is exactly wrapping around where the bacterial fission is happening. Now, the question is, how does it orchestrate? the small invagination which is created, now that invagination is because of the mitochondrial wrapping around of DRP1, is it a tripartite contact with STEA? That we are trying to understand, and we have not solved this mystery yet.
And my final question is, what signals the ER towards the vacuolar central and allows it to wrap?
What? It is all in the vicinity. If you really see a very enclosed, compartment, the tubules are longer. Now, why the tubules are longer? Because it induces the so-called ER stress. So you do see a sheet to tubule conversion, but it is always like a serendipity that why they are always in close positioning with the salmonella containing vacuole, right? So yes, it is all there, and that is how the STEA makes them to wrap around.
Okay,
Yeah.
thank you.
Thank you so much.
It was an interesting talk, ma'am. And I read one of your papers in which you mentioned sirtuins
Yeah,
and its connection with salmonella
Yeah.
and its pathogenesis.
yeah.
And I have read that sirtuins are also a part of hypertension. So, is there a possibility that the sirtuin, some of the protein has one part which is mechanically activated, which has some parts in the endoplasmic reticulum also, which makes it mechanistically sense the fission which happens in the salmonella, so that it senses and wraps around nicely.
Good thought. See, sirtuins are histone deacetylase, and normally it is not a part of hypertension, but it has a pleiotropic, lot of pleiotropic effect. Yeah, they play a role in diabetes, hypertension, so-called ventricular atrophy, etc., etc. So their knockout has a pleiotropic effect. Now, sirtuins also play a very global role in the metabolic flux of fatty acid to the glucogenesis or the glucose uptake and their further conversions. Sirtuins play that role. I have never thought from that angle that how it can play a role in the vacuolar division. But we know that from our experiments with sirtuins that it play a role by perturbing the metabolic flux. What it does, if the host flux is towards the glycolysis, it reduces the fatty acid oxidation in the bacteria and vice versa. But yes, it's a good thought process. I'll think over it. Thank you.